Today, I'll be talking about DNA replication. DNA replication has three stages. The first stage is initiation. DNA replication cannot begin without the unwinding of the double helix. For this, the enzyme helicase is required. It separates the DNA strands from each other by breaking the hydrogen bonds between base pairs. As you can see, the separated strands form a Y-shaped structure called the replication fork. Now, we need something to stabilize the fork structure. So, single-stranded binding proteins attach to the DNA strands to keep them separated. Now, the second stage of DNA replication is called DNA elongation. The two strands of the replication fork have a 3' end and a 5' end. The 3' end has the hydroxide group, whereas the 5' end has the phosphate group. As you can see, one of the strands is oriented in the 3 to 5' direction, whereas the other one is directed from 5 to 3'. Nucleotides are added to the 3' end because it is sticky. Moving on with the replication process, we only have one strand of DNA. To fully replicate the DNA, we need the second strand. This need is fulfilled by another enzyme called RNA primase. Primase builds a small RNA segment of about 10 to 60 ribonucleotide pieces of RNA called the RNA primer. RNA primer activates the enzyme called DNA polymerase III, which starts adding nucleotides to the 3' end of the RNA primer. So again, it replicates in the 5 to 3' direction. As a new strand is built, it is oriented in the opposite direction as the original or parent strand. Since the DNA polymerase III only builds in the 5 to 3' direction, this strand is built towards the replication fork, and as a result, the replication is continuous. This type of strand is called the leading strand. If we look at the second strand, obviously, it is oriented in the opposite direction as the first one, so the replication occurs away from the replication fork. In this strand, replication is a little different than in the leading strand. Instead of only one RNA primer, we need multiple primers and therefore multiple polymerase III activation. And this leads to the formation of short DNA fragments called the Okazaki fragments. Replication in the strand is not continuous and happens away from the fork, and this strand is called the lagging strand. So afterwards, another enzyme called DNA polymerase I removes ribonucleotides of the primer and replaces them with deoxyribonucleotides in the 5 to 3' direction. DNA replication wouldn't be done without obtaining the double helix, and in order to do this, we need to combine the gaps between two DNA strands as well as between the Okazaki fragments. The enzyme ligase is responsible for this. The last stage of DNA replication is termination. In this stage, RNA primer gets removed and we run into a problem because the 5' end of the chromosome is incomplete. It's left with some unpaired DNA nucleotides. The solution to this problem is telomeres, which are repeated sequence of TTAGG or TAG produced by telomerase and attached to the 5' end of the chromosome. DNA replication also looks for any errors or, in other words, mismatched base pairs. DNA polymerase III checks for the correct hydrogen bonds between nucleotide bases as they are put in place. When these do not occur correctly, the wrong nucleotide is removed and the correct one placed. At the end, we have two daughter DNAs. And that is the end of the DNA replication process.